everybody, welcome to your own classroom. Today in the fast track revision series we would be learning about generation and conduction of nerve impulse. For that we need to see the structure of neuron first. As you can see, a neuron has got a cell body, and cell body has got nucleus, Golgi apparatus, and there are several granules which are called as Nissel's granules present in the cell body. Actually, these granules are endoplasmic reticulum associated with the ribosomes, and it is the site of the protein synthesis. Then followed by cell body there is an axon, and it is the main site where the generation of nerve impulse takes place, and even the conduction happens here. And then there is an axon terminal, as you can see, and axon terminal has got several vesicles filled with neurotransmitters, and there are release sites here. So now let's start with the original process. As you can see, this diagram is showing exolemma. Exolemma is the membrane of an axon, and then there is exoplasm, that is the cytoplasm of the axon, and the outside of the exolemma has got positive charge and inside it is negatively charged. And this polarization is due to differential distribution of ions outside and inside the exoplasm. So let's see what ions are present on the outside. So on the outside there is a low concentration of potassium ion and high concentration of sodium ions, and inside the exoplasm there is very high concentration of potassium ions. That's why potassium is called as the most abundant intracellular cation. And then there are negatively charged proteins inside the exoplasm, and there is low concentration of sodium ions. And due to this differential distribution of ions across the exolemma, as we have seen, a concentration gradient is maintained in the resting nerve fiber. Means a nerve fiber that is not conducting an impulse, in that case also there is a potential difference. Now let's look at the factors which play a crucial role in the maintenance of this concentration gradient. This gradient is maintained by the active transport of ions by the sodium-potassium pump, which transports 3 sodium outwards and 2 potassium into the cell. So as you can see in this diagram over here, that this is a polarized membrane in the resting state. So this is a polarized membrane in the resting state, and there is a sodium potassium pump here which is sending 3 sodium ions outwards and 2 potassium ions inward. And due to this, in the resting stage, this concentration gradient is maintained. As a result, the outer surface has a positive charge and the inner surface becomes negatively charged. Means it is not that the inner surface has got only negative charge. There is a comparative thing here. As we have seen that inside the exoplasm there is high concentration of potassium ion, negatively charged protein, low sodium ions, and outside there is low potassium and high sodium ion. So outside there is higher concentration of positively charged ions, and inside there is higher concentration of negatively charged ion. That's why the polarity is that outside it is positive and inside it is negative. The electrical potential difference across the exolemma in resting nerve is called as resting membrane potential, and the state of resting membrane is called as polarized state. This state is called as polarized state. So when we keep an electrode inside the exolemma and one outside, then the potential difference comes out to be minus 70 millivolt, and this minus 70 millivolt is called as the resting membrane potential. Means the potential of the membrane when it is not conducting any impulse. Now if we compare the concentration of different ions outside the exoplasm, means in the extracellular matrix or extracellular fluid, and inside the exoplasm, it would be exoplasm, then you can see here the concentration of potassium ion outside the exoplasm is 5. These values are in millimole. So the concentration of potassium ion is 5 and inside it is 150 outside the concentration of sodium is 145 and inside it is 15. The concentration of calcium outside it is 2 and inside it is 0.0002, and the concentration of chloride outside is 125 and inside it is 9. So this is just showing a comparative data of the distribution of different ions. Now I think I have missed one thing in the previous diagram. As you can see here, let me show it with a pointer. I have shown that there is greater movement of potassium ion outside the cells and less movement of sodium ion inside the cell. Means these ions have a tendency to move from their higher concentration to the lower concentration. So as we have seen, the potassium ions have a higher concentration inside the cells, so they will move outside, and sodium ions are in higher concentration on the outside, so they will move inside. This is clear, but even in the resting stage there is a continuous movement of ions, and this movement is allowed by the leak channels. So let's see what are leak channels first. So it is written here that at resting stage membrane is more permeable to potassium ion and less to the sodium ion and least to the negatively charged proteins. This movement is maintained by the leak channels, which always remain open. So there are basically three types of channels. First is leak channel. These are ionic channels that remain open all the time, even in the resting stage. Then there are ligand gated channels. Ligand gated channels are chemically gated channels which open or close in response to chemical stimuli. So if you remember the synaptic cleft or neuromuscular junction, you have seen that when acetylcholine is released from the presynaptic neuron, it is accepted by the postsynaptic neuron through the receptors present on the postsynaptic neuron, leading to the opening of sodium channels in the postsynaptic neuron. So this was an example of ligand gated channel, when binding of a ligand opens the channel. And then there is a voltage gated channel, and these are mechanically gated channels which open in response to a physical stimulus in the form of vibration such as touch and pressure. So this is what we are going to see next. All or none principle. The strength by which a nerve or muscle fiber responds to a stimulus is independent of the strength of stimulus. It means that no matter how much stimulus we give, if it is not of the threshold value, then the muscle or the nerve fiber won't respond. And if that stimulus exceeds the threshold potential, the nerve or muscle fiber will give a complete response. So this is all or none principle, and each stimulus of threshold value causes same amount of voltage change in the neuron. It disrupts the polarization of the membrane, leading to a process called as depolarization. So that's what we are going to study now. For most excitable cell, the threshold potential is -55 to -60 millivolt. This is very important. Means this is 10 millivolts higher than the resting membrane potential. Nearly 10 millivolts. Now as you can see, this is the diagram of a first let's look at this part. So as you can see, this part is showing a polarized membrane. That's what we have seen in the resting membrane condition, that inside it is negatively charged and outside there is a positive charge. And then if this membrane receives a stimulus, so if the stimulus is being applied from here, then sodium voltage gated channels open in the region of the stimulus, and there is a large influx of the sodium in this region. And due to the entry of sodium in this region, due to the large influx of this sodium in this region, there would be an increase in the positive charge, and this positive charge would lead to the change of the polarization in this particular region. Inside it would become positive and outside it would become negative, and this is said to be depolarized. And depolarization is mainly due to the opening of voltage gated sodium channels, but voltage gated potassium channels are closed at this point. So what all happens during a depolarization event? When a threshold stimulus is being applied to the polarized membrane, the membrane's permeability to sodium ion increases at that point of stimulation, means at the point of application of the stimulus. It is due to the opening of voltage gated sodium channel and potassium channel remain closed. Sodium channel influx takes place from a region of its higher concentration to a region of lower concentration. The electrical potential of the membrane changes from minus 70 millivolts towards the 0 millivolt, means the potential is going towards the plus side or positive side, and the membrane is said to be depolarized. Throughout depolarization, the sodium ions rush inside the membrane until the membrane becomes negatively charged outside and the positively charged inside, and the potential becomes plus 30 millivolts. Means the depolarization continues till the potential of the membrane changes from minus 70 millivolt to plus 30 millivolt, and a condition like this is seen. Now let's move to the next slide, and it is showing repolarization. So once the membrane is depolarized, means the potential is changed, then there is a time for repolarization, means for the reversal of the polarity. So what happens at the point of repolarization? The voltage gated potassium channel opens and a large amount of potassium ions move from inside to the outside, leading to the change in polarity to the original polarity as we have seen in the resting condition, and the polarity is reversed. That's why it is called as repolarization. Membrane is again in the polarized state. So this region is showing the depolarized region, and this is the original actual polarized membrane region, and this is showing the repolarized condition when voltage gated potassium channel opens and a large amount of potassium is sent outside from the inside, leading to the change in the polarity to the like the polarized membrane. Now let's look at the another slide. So what all happens in repolarization, let's see this in detail. So in repolarization, with increase in sodium ions inside the nerve fibers, so the first point is basically talking about depolarization. With increase in the sodium ions inside the nerve fibers, the membrane becomes less permeable to sodium ions and more to the potassium ions. Means when the membrane has reached plus 30 millivolt, it's the time that there should be less sodium influx into the cells, and the sodium channel close and the potassium voltage gated channel starts widening, means starts opening, and due to the opening of this voltage gated potassium channels, potassium outflow begins and the resting potential is restored, means inside it becomes negative and outside it becomes positive. Now we will talk about action potential. So what is action potential? Any change in the electrical potential associated with the passage of impulse in the nerve is called as action potential. Means whenever a impulse is passing through a nerve, then whatever change is created by that impulse in terms of the electrical potential change, that is called as action potential. And whatever events that we have studied till now, like depolarization, repolarization, all are the part of the action potential. It is also referred as nerve impulse or spike. Now let's look at this curve. On the y-axis, as you can see, voltage is there in millivolts, and on the x-axis time is given in milliseconds. Now this is minus 70 millivolt, this is the resting membrane potential when no impulse is generated, and at this time this minus 70 millivolt is maintained by the sodium potassium ATPase pump. If you remember, Now when a stimulus of a threshold value is given, means that stimulus is of 10 millivolt higher than the resting membrane potential, means it is of minus 55 to minus 60 millivolt, then an action potential is generated, means it will lead to the opening of the voltage gated sodium channels and depolarization would occur, and the membrane potential starts changing from minus 70 towards 0 and finally to the plus 40 or plus 30 millivolt. At the plus 40 or plus 30 millivolt, all the sodium channels close and all the potassium voltage gated channel open, and this will lead to the efflux, means outflow of the potassium from the exoplasm to the outside, leading to the repolarization of the membrane. But there is one problem with the potassium voltage gated channel, that it does not close at the resting membrane potential, means the channel should have closed here, but it goes below this, and this potassium channel close at minus 80 to minus 90 millivolt as it is written here, and this extra dip below the resting membrane potential is referred as hyperpolarization. So this state is referred as hyperpolarization, means potassium channels remain open for a longer time, and to bring back this to a normal or a resting state, again sodium potassium pump is required. So let's see it with a clearer graph. Here, as you can see, this is the threshold potential minus 55 millivolt, this is the resting membrane potential minus 70 millivolt, and this is showing that dip or the time for which the potassium channels remain open for a longer time, and this is referred as hyperpolarization. Then there is something called as refractory period. Refractory period is that period during which no matter how much stimulus you apply, a second action potential won't be generated. So this curve, what I am showing here, is action potential. So what is that time? Let's see. As you can see here, this time period means the period from which is covering depolarization and 2/3 of the repolarization is called as absolute refractory period, and this is referred as relative refractory period, means last 1/3 part of the repolarization. This is called as relative refractory period, means during absolute refractory period, no matter how much stimulus you give, a second action potential absolutely can't be generated. And during the relative refractory period, if you give a stimulus above the threshold value, then a second action potential could be generated. So this is the concept of refractory period. Now let's look at some major points here. Why repolarization is necessary? Because it prepares the nerve fiber to receive another stimulus and conduct it in the same manner. Second, the nerve can transmit about 1000 impulses per second at a maximum speed of 130 meters per second. During refractory period, the membrane is incapable of generating another impulse. We have already seen this, and refractory period ensures unidirectional impulse conduction in the axon. So it ensures impulse conduction in one direction. Now let's look at another slide. It is regarding the conduction of impulse. So we have already seen so much about the generation of the impulse. Now let's talk about the conduction of the impulse. So far we have been talking about the non-myelinated neuron, where you have seen that inside it is negatively charged and outside it is positive charge in the resting condition. So in non-myelinated neuron, the stimulated negatively charged point on the outside of the membrane, means when a stimulus is given to a resting neuron, what would happen? Sodium channels open at the site of the stimulus, leading to the change in polarity. Outside it becomes negative and inside it becomes positive. So this negative charge point on the outside of the membrane sends out an electrical current to the positive charged point on the uh, point that is adjacent to it. Means this negative charged point on the outside sends out a electrical current to the positive charge point adjacent to it. In this diagram you can see the arrows. These arrows are denoting the flow of electrons, and as you know, the flow of an electron is of the opposite direction of the flow of the current. So this statement is correct. It says that the negatively charged point on the outside of the membrane sends an electrical current to the positive point that is still polarized. This local current causes adjacent inner part of the membrane to reverse its potential from the -70 to +30 millivolt. So that is what is seen during depolarization. Means now at one point, this on giving the stimulus, if this occurs or if this happens, then this local circuit propagates throughout the length of the neuron, changing the polarity, and then the membrane is said to be depolarized, and polarity changes from -70 to -30 millivolt. And this reversal repeats itself over and over until the nerve impulse is conducted throughout the length of the neuron. So in case of myelinated neuron, the speed of conduction is 2 meter per second. And now let's talk about the unmyelinated neuron, or how the conduction takes place in the unmyelinated neuron. So depolarization occurs only at the nodes of Ranvier. If you have seen unmyelinated neuron, you know that in unmyelinated neuron, myelin sheath or medullary sheath are present, and these myelin sheath and medullary sheath means between two myelin sheath there is a region called as node of Ranvier, and this is the region where the concentration of ion channels are very high, and the electrical potential generation takes place at this point, and this is via leaping or the jumping of the potential. And how it takes place, let's understand. Suppose once we have given the stimulus at this point, the polarity is changed and the current flows from the terminals, and a local circuit is created here, and now this local circuit jumps, and this is comparatively faster to the in comparison to the non-myelinated neuron. The speed of the conduction is 120 meter per second. So it leaps over myelin sheath from one node of Ranvier to another node of Ranvier. And this kind of conduction is called as saltatory. Saltaire means to jump. Now let's look at a summary of everything, whatever we have seen so far. In the next slide. So this is a very good diagram which is showing everything whatever we have seen. At the first, or in the first diagram, as you can see, voltage gated sodium channel is in resting stage and voltage gated potassium channel is closed. Means both the channels are closed at this point. Both the channels are closed. Now once a stimulus is given, sodium channel opens and sodium flows inwards. As you can see, the sodium channel opens and the sodium flows inwards, and leading to a process called as depolarization. Now once depolarization starts, you can also see the change in the action potential in this small curve over here. So depolarization has just begun. Now once depolarization is the potential of the membrane has crossed or has reached +30 millivolt, then repolarization begins and potassium channel opens and sodium channel gated channel closed. So as you can see, sodium voltage gated channel closes here and the potassium voltage gated channel opens, leading to the outward flow of the potassium, and the curve takes this form. Now let's look at another event. This is showing the completion of the repolarization. when the potassium channel exits and sodium channel begins to open again, and repolarization completes here, and a resting potential is restored. So this is all in this. Thank you.